Good afternoon, everyone. Myself, Dr. P. Samuel, Assistant Professor, Department of Biotechnology, Bishop Heber College, Tiruchirappalli. In this video tutorial, we'll be going to discuss about transformation in bacteria. So, in the previous presentation, we discussed about conjugation, a kind of horizontal gene transfer between bacterial partners through which the recipient acquire new traits. In the same way, transformation is also called as a horizontal gene transfer method by which a bacteria takes up the DNA from the ecosystem, from the environment in which it is growing. So let us discuss the process in detail in the upcoming slides. Let me go to the first slide. We all know that every bacteria has a prominent cell wall which is acting as a boundary protecting the cell from any devastating conditions such as changes in temperature, changes in pressure, changes in chemicals around the ecosystem, right? All these things are potent um, agents that destroy a bacterial cell. But from all these things, cell wall is a major factor that helps the bacteria to overcome all the adverse condition. So within the cell wall, let me explain it. The outer blue color border is called cell wall. I refer it as CW. And this layer, this transparent layer is said to be this one is said to be the cytoplasm, right? Cytoplasm, okay, right. And the condensed mass in the cytoplasm is said to be this area. This is said to be the chromosomal DNA of the bacterium, right? So a typical bacteria has a cell wall, cytoplasm, and in the cytoplasm you could find the genetic material which is assembled in the form of tangled mass, right? So under unfavorable condition, a cell lose its viability, right? So the loss of viability is mentioned here by the formation of pores in the cell wall, right? Pore 1, right? Pore 2, like this multiple pores will be formed, P4, right? P5, multiple pores will be formed under unfavorable conditions, or when the condition or when the environment is not suitable for the organism, for the bacteria, the cell started to lose its viability, the cell started to die, right? And this is indicated by the formation of multiple pores on the cell wall. Next slide. And through the pores, right, in the previous slide, through the pores, here you could find the chromosomal DNA which is organized in the cytoplasm, right? Or in short it is called as chromosomal DNA. And this chromosomal DNA will be fragmented upon cell lysis, and these fragmented DNA will comes into the environment upon the cell is eventually dying, right? So these are lysed DNA fragments released from the dead cell, right? These are lysed DNA fragments. All these red color lines are referred to as lysed DNA fragments released by the dead cell. Imagine when the same ecosystem is also favoring the growth of some other bacteria, right? See in the previous slide, we know that due to adverse conditions, there are some cells, there are some bacterial populations are dying. But in the same ecosystem, there are some other cells are living because of their nature, right? And in the cells, under some circumstances called competent stage, mark my words, the stage is called competent stage, COMPETENT, competent stage. So this is the stage not seen in the bacteria at all time. Only during the particular time interval, a cell develop the stage of competency, right? For example, Streptococcus pneumoniae, Bacillus subtilis are able to take up the DNA directly from the ecosystem. And how these DNA are taken up into the cell, that is the most interesting point. So we'll discuss about it in the next slide. See, during the stage of competence, bacterial cell develop some special proteins which are not seen in the bacteria at all time. These proteins are called as transformation bud. Transformation bud. In some books, it is referred to as transformosome. Transformosome. Remember these keywords: transformosome. Added, remember these transformosome are not expressed at all times. Only during the stage of competency, cells like Streptococcus pneumoniae or Bacillus subtilis are expressing these transformosome protein on the cell wall. So what is the role of this transformosome protein? And these proteins are specialized receptors, right, through which the DNA is taken up by the cell. I told you this is a receptor through which the lysed DNA are transported into the cell, right? And like the process continues, right? Here you could see the transfer of or the uptake of genetic material into the bacterial cell through the transformosome bud or the receptor protein expressed on the cell during the stage of competency, right? So upon the cell or the DNA bounds to the transformosome bud or transformosome protein, it is gradually taken up into the cell. The lysed DNA is gradually taken into the cell, right? Remember, this is the stage in which the DNA, I mean the lysed DNA, found to be associated with the transformation bud. In the next slide, the DNA is completely transported into the cell, leaving behind the transformation bud empty, right? So upon successful transfer of the DNA from the external environment through the internal environment of the cell, through the transformosome bud, right? Through the transformosome bud, upon successful transfer, the transformation bud started to disappear. See, here you couldn't find any transformosome bud or transformation bud on the cell wall. So it will be disappeared, which means what? It lasts for few minutes or few time, right? After that, the transformation bud started to disappear. Once the DNA get into the cell, right, the DNA started to integrate. So where it undergoes integration, the integration takes place in the chromosomal DNA, either here or some other places, right? And how this recombination takes place and how this binding, because this is an exogenous DNA, right? Exogenous DNA is the DNA released by some other cell, right? Now the cell of non-indigenous bacteria enter into a new bacterium through the transformation bud, everything, right? After entry of the DNA into the new cell, the DNA finds a place to recombination, right? So this process is called homologous recombination, which enable the DNA to bind with the chromosomal DNA, right? The process is called integration. It is facilitated by an enzyme called integrase, right? Upon integration, see, this is the place where the newly entered DNA recombined with the bacterial chromosomal DNA, right? This is the site of integration, and the process of integration is facilitated by integrase enzyme. You are supposed to remember all the key enzymes playing a major role in the entire process. This is very essential or important things to be noted by a student, right? So remember, this is the cell wall, the outermost layer. Here you find cell wall, right? So upon integration, the bacteria started to show, right, some new properties, right? Upon integration of exogenous DNA into the chromosomal DNA, the organism started to show new properties, right? What is the new property? See, the formation of capsule. Originally, the organism is lacking capsule, but upon integration of the exogenous DNA, the recipient organism started to form capsule across around the cell wall. This is an additional feature that provides protection to the organism, and it also makes the organism resistant to phagocytosis. In this way, the bacteria acquire new properties. For example, antibiotic resistance, capsule formation. So all those new properties are acquired by the bacterium through the horizontal gene transfer methods like either conjugation or transformation steps. So that's all for now. If you have any queries, please don't hesitate. Just shoot your questions to me. I'm available to clarify your doubts. Stay tuned. Bye bye.